Okay, thank you very much for joining today's UCI Mind Seminar. It's a great honor to introduce today's seminar speaker, Dr. Pinar Ayata from the City University of New York. Before starting her seminar today, I'd like to introduce her background very briefly. Dr. Ayata received the Ph.D. degrees at the Rockefeller University under the mentorship of the Dr. Nathaniel Haynes, where she worked on the some epigenetic modification and epigenetic related study and Rett syndrome. After she received the PhD, she moved to the Mount Sinai and did the postdoctoral work under the mentorship of Dr. Ann Schaefer, who is an leading expert of the microglia biology in the field. And she worked on the very interesting study using the very nice techniques and transcript techniques and isolating the particular subtypes of microglia and functionally specialize the phagocytic clearance of the apoptotic neurons. Her work has been highlighted very highly in the epigenetic mechanisms to maintain specialized specification of microglia importance in the normal brain function. She has been publishing the very top tier journals, including Nature Neuroscience in 2018 and Nature in 2020, just last year. Also, she has received numbers of fellowships and awards during her postdoctoral work at the Mount Sinai, including the Science Graduate Fellow Award, Rockefeller. This is an award from the Rockefeller University during her graduate work, and also the NRSAD Young Investigator Award, Robin Clemens Postdoctoral Award, and so on. Now, after completion of her postdoctoral training, she recently moved to the City University of New York, where she is holding assistant professor over there. Actually moved last year, August 2020. So her lab is very new, and during the pandemic, it's been a difficult time, but I guess she's establishing her independent work there and continue work on the understanding the specialized function of the microglia subpopulation in the health and disease brains. So without any further ado, I'd like to welcome Dr. Pinar Ayata today's talk. Her seminar title is Functional Specialization Microglia in Health and Disease. Dr. Ayata.
Thank you so much, Masajid. I'm really honored for your invitation. So I will test if you can see my slides. Fine.
Yes.
Hope so. Okay, great. As Masashi said, I recently started my lab at this new institute that CUNY started. It's called Advanced Science Research Center, or ASRC. It's a very young institute with a very interdisciplinary approach. But today I will mostly be talking about my postdoctoral work focused on understanding microglia subtypes in healthy brain as well as in during the neurodegeneration. So basically, I will talk about, I separated this in three parts. First, I will tell you a little bit about my published work, discussing the microglia subtypes in the healthy brain. Then I will move to neurodegeneration, which is unpublished results. And in part three, I will briefly mention what I'll be studying in my own lab. So our brain has two major cell types, as you all know, and the principal cells of the brain are neurons that perform the primary function of the brain, which is to transfer electrical signals from one functional region to another one. And the health and function of neurons are supported by another cell type called glia, and these cell types perform critical functions. Among these cells, microglia are a unique population. One of the uniqueness is their origin. So these cells, unlike the other types of glia originate from the yolk sac, and they come from myeloid progenitors. And once the blood-brain barrier closes, they maintain themselves as a self-renewing population, and they're very long-lived cells. Um, Microglia, as you can see, are highly plastic cells, and they can respond to this, for instance, a laser injury that was just introduced in a very speedy fashion, become activated, they proliferate, migrate toward the injury site, they change their morphology. And because of their properties that were discovered early on, they were thought to be this dormant cells that don't do a lot of that don't perform a lot of functions in healthy brain, but mostly are involved when there's an injury or pathology. So they were discovered about 100 years ago, but in the last couple decades, we are actually really starting to understand what they're doing. And this really changed with this particular study that I'm showing you in the video, where scientists were able to visualize microglia in the live brain. And what they saw, they wanted to study how they respond to pathology, and they did. But what they also saw was that microglia before pathology look nothing like dormant. They seem to be highly dynamically interacting with the tissue in their vicinity, which this finding really sparked the modern field of microglia. And today we know that microglia perform critical functions such as genetic macrophage functions, clearing debris, or immune surveillance that are shared with other tissue macrophages, or they also perform brain tissue specific functions, such as assisting neurogenesis, synaptic remodeling, secretion of neurotrophic factors, and modulation of neural activity. They can respond to the needs of their environment quite fast. And usually they can enter from functional state to another functional state. And this is reflected in their morphology and associated with a unique transcriptional program. So when I started as a postdoc, one of our main questions was, okay, if tissue macrophages specialize to each of the tissue they reside. Now, if you look at the brain, it consists of functionally and structurally specialized regions. So you can almost consider each brain region as a unique organ. So what we wondered is if microglia specialize in these different brain regions, because the needs of each brain region containing different neuron subtypes will be different. And in fact, when we just look at how microglia look in these different brain regions, you can see that, you can appreciate that they look quite different. For instance, if we focus on the two really differently looking brain regions, for instance, striatum and cerebellum, you can see that in the striatum, microglia look much more branched, which is usually associated with a dynamically interacting state. On the other hand, cerebellar microglia are not only less ramified, but they also contain a higher number of lysosomes, suggesting that they might have heightened clearance activity. So based on this initial observation, we were really interested on answering these questions on region-specific differences of microglia. For instance, do microglia have region-specific differences at the molecular and functional level? And what is upstream of this specification? And what's the functional output of this specification? So we first wanted to understand, so what are the differences at the molecular level? So for this, we, of course, our first target is to look at the transcriptome of these cells. Traditionally, to get microglial transcriptome involves their isolation from the brain tissue and then obtaining their RNA basically. But what if you just remember the movie I just showed you. A small tissue injury, a very focused laser-induced tissue injury is enough to activate, um, several microglia around them within minutes. So if you imagine this, let's say, enzyme-induced digestion of the tissue, homogenization, all of these methods will appear to microglia as a tissue injury. And then once you have isolated microglia, they have lost all homeostatic signals that make them themselves. And they also are activated from all the injury signals around them. So this will result, we predicted that this would result in this artificially activated state. So to prevent this, we used a method, we adopted a method that was developed to obtain neuronal transcriptome. And this method is called TRAP, or translating ribosome affinity purification. In this method, we genetically label ribosomes of a particular cell type with GFP, and this allows us to digest the tissue and the cells at the same time, and purify GFP labeled ribosomes, which are found in polysomes. Purify these GFP containing polysomes using anti-GFP antibodies, and this way we only obtain mRNA from this specific cell type. So I first applied this method to microglia. Oh, sorry. Here in the original paper from 2008, you can see an electron microscopy image of a magnetic bead that is bound to these polysomes that I was describing. So I first adopted this technology to microglia by screening a number of different lines, and we found that CXCR1 tamoxifen-inducible Cre line was really beautifully targeting microglia. And you can see in this image that microglial ribosomes are in a nucleolus, in the cytosol, as well as at the branching points of the protrusions, which is very interesting. And the other advantage of TRAP is that it not only provides you the transcriptome, but it rather provides the translitome of a cell, which is closer to its proteome. And also, because the ribosomes are assembled in the nucleolus, the TRAP mice allow us a label-free method to isolate nuclei based on their GFP expression. So using this mice, we first compared these different methods. We used the same mouse, and we either performed TRAP, the isolation of ribosomes. We compared this with the nuclear isolation of microglial nuclear isolation, and also we performed the whole cell isolation of microglia. And only when we isolated whole cells, we obtained this artificial activation signature consisting of immediate early genes, pro-inflammatory genes, and stress response genes, indicating that indeed isolation of whole cells allow this reactivation of microglia in an artificial fashion. And we use this TRAP technology to profile striatal and cerebellar microglia, where we initially saw ramified versus less ramified and high in lysosome content morphologies. And what we predicted back upon seeing those morphological features was that striatal microglia would be more in surveillance mode, whereas cerebellar microglia would be more in the phagocytic clearance mode. And these functional states were reflected in their transcriptome as well. We observed more clearance genes in the cerebellar microglia, and more surveillance, motility, chemotaxis, cytokine secretion, and so on. This genes that are associated with these functions in the forebrain microglia. We also use the single nuclei sequencing and confirm these signatures at the single cell level. So Now, when we compare these signatures, what we immediately noticed is that cerebellar microglia signature had a lot of similarities to this microglia associated with neurodegeneration. This signature is conserved in AD, aging, or ALS. It had also similarities with microglia associated with neurodevelopmental. And what these two temporal phases have in common is that there's a neuronal loss or neuronal attrition. So what we wanted to know is that is there neuronal loss in the cerebellum as well, which could be the external signal driving this phagocytic signature. So our first clue came from our morphological analysis where we were looking for quantifying lysosomes. What we again and again started to notice was that these lysosomal particles would contain pieces of DAPI, areas of DAPI positive particles, which indicated that these microglia have eaten a whole cell rather than myelin or synapses or spines, neuronal spines, and so on. So, but when we looked at the brain, we don't really see dying neurons in any brain regions. Then we went back to the literature, and what is common is that if there's an apoptotic cell undergoing apoptosis, um, phagocytes around this cell will remove it before our technologies can detect apoptotic cells. So what we needed to do was to prevent the phagocytosis, the removal of these apoptotic cells. So for this, we use two methods. One is a pharmacological method to ablate microglia in the whole brain, and another use a genetic model to prevent phagocytosis. And using both methods, we were able to reveal a high number of apoptotic cells in the cerebellum, but not in the striatum, which meant that cerebellum microglia are continuously exposed to these apoptotic cells, whereas forebrain microglia are not, so they can specialize in other functions. Our next question was, do cerebellar microglia specialize in phagocytosis since their molecular identity indicates this? And for this, to answer this, we did two different experiments, two complementary experiments. One to test phagocytic efficiency. One was ex vivo, where we isolated microglia from cerebellum and striatum, cultured them briefly until they attached the plate, and then we exposed them to apoptotic cells and then assessed the efficiency of phagocytosis by flow cytometry. We complemented this by an in vivo approach done in Oleg Butowski's lab, where they injected early apoptotic neurons in these two brain regions, then isolated microglia 3 hours later, and again analyzed by flow cytometry. And these two independent approaches resulted in very similar differences in efficiency, where cerebellar microglia are 2 to 3-fold better at removing apoptotic cells, which means that they're already functionally specialized. They're more ready to remove apoptotic cells. Next, after showing that these differences at molecular, morphological, and functional level, our next question was, okay, but what is driving these differences, and what is the molecular mechanism that is driving this? As I told you earlier, there's a difference in the region, and one of the big differences is that cerebellar microglia are exposed to these apoptotic neurons. So we asked, can this be driven by the exposure to apoptotic neurons? Can the phagocytic signature be driven by exposure? And for this, we first exposed cultured microglia to apoptotic cells, and we showed that this resulted in induction of genes associated with clearance, such as engulfment, lysosomes, and lipid metabolism. What we also noticed was that the same exposure induced regulators of H3K27 trimethylation, which then led us to ask if this could be a mechanism where exposure to apoptotic cells could drive the phagocytic signature. So to step back and talk about a little bit about epigenetics, our DNA is wrapped around these protein complexes called histones, and each histone has a tail, unstructured tail that is sticking out of this complex. And these tails can be post-translationally modified. And each post-translational modification will lead to either compaction of the DNA around that area or increase in accessibility. So modifications that increase accessibility will lead to activation of genes, and modifications that result in compaction will lead to repression of the genes. So what we found is the mechanism that involves the trimethylation of lysine number 27 on histone H3. And this is usually, this is associated with repression of genes. And this mark is placed by a complex called PRC2, or polycomb repressive complex 2, and it is erased by KDM6A and B. And what we found was that exposure to apoptotic neurons induces the activity of KDM6s. And what else is important about this mark is that it is involved in cell subtype specification, as Anne Schaefer's lab previously had shown. And it is also thought to be involved in macrophage polarization. So this was a great potential mechanism for us to explore further and ask, is this a mechanism necessary to maintain the subtype identities of microglia? So first we wanted to see, are there differences in the genes that are repressed by H3K27 trimethylation between cerebellum and striatum microglia? And what we found is that indeed in the forebrain microglia, we see more of this repressive mark on clearance-associated genes, repressing their expression. And in the cerebellar microglia, we see increased presence of this mark on surveillance-associated genes, repressing their expression and pushing the cell more toward a clearance state. But of course, this correlation doesn't mean that this is the mechanism that maintains the region specificity of microglia. So we wanted to functionally test this. And to achieve this, we wanted to remove the mark from the system. And to do that, we looked at polycomb repressive 2, which is made of which has 3 major subunits. It has more, but the essential subunits are these 3. And loss of either subunit will result in the loss of function of the complex. So what we did was to delete the subunit called EED, and that really resulted in the complete loss of the complex, as well as H3K27 trimethylation signal. Here we can see a Western blot of microglial nuclei from these mice. And at the bottom, you can see microglia in green and H3K27 trimethylation mark in red. And you can see that in the knockout animals, we only removed this mark from microglia. And what we did after that was to look at microglia identity over time. And what we saw, for instance, in the forebrain microglia was that we have a time-dependent loss of these surveillance-associated genes, which are the characteristic of forebrain microglia. And at the same time, we have an increase in the phagocytic genes. And these changes were also reflected in morphological features of microglia, where we see a loss of ramification and increase in lysosomes, similar to those in the cerebellum microglia. So then we wanted to ask, does this then affect brain function in any way? And to test this, we first looked at neurons. So assuming that microglia in the stratum are specialized to serve the needs of their neighboring neurons, now they have a different identity. Does this result in does this affect the um, health of the neurons around them? And what we saw was indeed that there's a loss of spine density in these neurons in this region, indicating that microglia preservation of microglia identity is necessary um, to for the health of the neuron. And I'm just going to summarize it with two words, a few words. I can't see it. But we also ran a battery of behavioral experiments, and what we observed was that as there's a time-dependent loss of behavioral fitness that is that was evident in their in neuronal sensitivity, in anxiety-like behaviors, in cognitive performance, and we also observed a handling in induced seizures, occurrence of handling induced seizures. Um, I am actually showing it. I thought I removed the slide. I apologize. So here are some of the behaviors we tested, and we see that the behavioral output is really dependent on microglial identity. So to summarize this section, we identified that microglia in striatum and cerebellum have diverse identities, and in the cerebellum, we have a rather phagocytic phenotype, which is associated with apoptotic neurons specific to the cerebellum. And this is a partially adaptive phenotype. And these differences are maintained by HGK27 trimethylation epigenetic mark. And loss of this and maintenance of these different identities is important for normal brain function. So now I'm going to switch gears and talk about a little bit about unpublished work. As we were uncovering our findings about microglia, what we immediately noticed in the newly coming studies was that microglia in neurodegenerative diseases had a very similar phenotype to our phagocytic cerebellar microglia. And a very recent study beautifully shows what many studies have found before, showing that the genes that influence AD risk are mainly on the enhancers of microglia, which means that epigenetic regulation in microglia is really a major mechanism that influences the risk for AD. So if we step back and think about AD, what is AD and what are the pathologies of AD? So there are two pathologies in tissue. One is amyloid plaques and the other one is tau. And there were years-long debates about what matters more, but now the field seems to be accepting more and more that the amyloid plaques are the trigger of the disease, because these are the primary pathology, and you can have plaques for decades and you won't necessarily develop dementia. Whereas tau aggregates appear later and they precede neurodegeneration and cognitive decline, which resulted some people calling the amyloid plaques as a trigger and the tau as a bullet. So what role microglia might be playing in this? So one of the most studied microglia subtypes in Alzheimer's disease is this plaque-associated microglia. It's also it's also a characteristic of this microgliosis, where microglia show this activated, polarized morphology around these amyloid plaques. But what an other AD-associated microglia phenotype that is identified is this aberrantly activated phenotype. It could be, people have called it different names, but it's a different microglia subtype that is morphologically different, and the emergence of these microglia precede tau pathology, which precedes neurodegeneration and cognitive decline, which resulted in kind of people suggesting that these might be the harmful population, whereas the plaque-associated microglia might be more protective population. And in this case, they could be considered as the weapon in this analogy. So what we wanted to understand was that because there's this dichotomy about microglia, are they protective? Are they harmful? What we wanted to know was that can we separate the protective and harmful microglia populations from each other, and what are their identities? So for this, we again formed our questions. How can we differentiate them? What is their identity? What is the molecular mechanism regulating these populations? And what is the outcome what is their functional outcome in AD pathology? So how can we differentiate this? So thankfully there were a few studies, and in our hands, what we discovered, and multiple studies also discovered, was when people were looking at what do microglia do in Alzheimer's disease, and what do you do if you just want to study a cell, the role of a cell type? You can just delete it, and we have a drug for that. So multiple groups deleted microglia, ablated microglia, and looked at the disease outcome. But what we all observed was kind of unexpected, and it led us to suggest that maybe instead of trying to find what's different, how can we genetically target these microglia populations, we can just kill one and study all microglia, all remaining microglia, and then kill the other and study all remaining microglia. So microglia survival is dependent on the signaling through this receptor called CSF1R. If you block CSF1R, you kill almost all microglia. About 98% of microglia are gone. In 5XFAD, you have these different types of microglia. And if you give them an inhibitor, what happens is that you lose all these non-plaque associated microglia, but some of the plaque associated microglia remain. So at the same time, if you inhibit phagocytic receptor signaling through, for instance, TREM2 knockouts, or now we know in axl merTK knockouts, what happens is that, and Marco Colonna's group nicely shows this here, is that you lose microglia around the plaques. So now this gives us a method to use CSF1R inhibitor to kill all microglia but plaque-associated microglia, or kill phagocytic microglia or plaque-associated microglia and leave the rest of the microglia there. And from the literature that did this, that studied these different mice, we know that CSF1R inhibition has largely protective consequences, and inhibition of TREM2 and axl merTK has largely harmful consequences. So we went one step further and then asked which kinases or which signaling molecules could be downstream of this phagocytic receptor signaling. So we first blocked CSF1R signaling using CSF1R inhibitor. And then we blocked two different kinases that are predicted to be downstream of TREM2. And what we found was that only if we use PLC inhibitor, there's a significant decrease in the microglia populations. And we also showed using a genetic knockout, microglia-specific genetic knockout of PLC signaling, we show that indeed all microglia are gone if we use a genetic model telling us that PLC, PLC gamma 2 dependent phagocytic receptor signaling is really an alternative survival mechanism for these plaque associated microglia. So now, using this method, we were able to perform TRAP and ask what is the unique signature that we observe only in 5xFAD, but we don't observe in this, and what is the unique signature from 5xFAD, which is the Alzheimer's disease model with CSF1R inhibition, and then compared to the wild type. And what we found using this comparative study was that plaque-associated microglia rather express this innate immune response genes, which can be interferon response, MHC2, cytokine release, and also or complement pathway, and also phagocytosis, such as lysosome, lipid metabolism, and so on. And on the other hand, we find this in aberrantly activated or CSF1R dependent AD-associated microglia. We find this other signature that is rather global signature, which is associated with metabolic dysfunction and stress response. And this involves RNA decay, loss of translation machinery, and DNA damage response. And when we correlated this with Alison Goetz' group with AD risk genes, we found that plaque-associated microglia are rather correlated with reduced AD risk, whereas the aberrantly activated microglia have a higher correlation with increased AD risk, suggesting that these are really the harmful population. So then we ask, what is upstream of these specification of microglia? So looking at the upstream regulators of these different microglia subtypes, what we noticed was that this transcription factor called SPI1 comes up over and over again. That can activate or suppress genes and is found as activated in these comparative studies. Now, SPI1 is a myeloid lineage determining transcription factor, both for myeloid cells actually and B cells, and its expression level actually can define the identity of the cell from the hematopoietic state. And you can actually give, so lower expression of SPI1 makes a B cell, and higher expression makes a macrophage, which is the origin of microglia. And you can, in fact, increase the SPI1 expression in a pro-B cell and make a macrophage from a B cell. Moreover, B cell subtypes are also dependent on SPI1 levels, and you can manipulate, you can change the subtype of a B cell by changing SPI1 levels. So what we wanted to then ask was that, are the SPI1 levels, could SPI1 levels be determining the subtype identity of microglia? And we then looked, okay, so how is the expression of SPI1 in microglia associated with Alzheimer's disease? So what you can immediately appreciate here is that there's a very high heterogeneity of P1 levels that we observe over and over again, and this is especially clear here around the plaque-associated microglia. We have some that are really blasting with P1, in green, and others that are low in P1. Now, what happens if we give CSF1R inhibitor to these mice is that we only have these lower expression of, we have these microglia left that only express lower levels of P1, which indicated to us that it is possible that lower expression of P1 could be pushing microglia toward this plaque-associated protective subtype, whereas higher expression could be pushing them toward this aberrantly activated phenotype. More importantly, Alison Goetz' group recently found that lower expression of SPI1 is associated with lower AD risk, and higher expression is associated with higher AD risk. So what we wanted to know is that, can we modulate SPI1 expression to push microglia toward one or the other subtype? And I just want to also mention that AD heritability is also enriched in P1 cystrom or P1 target genes, which means that P1 could be considered as a master regulator for AD risk. So to answer our question, we made mouse lines where we either knock down or knock out a single allele of SPI1 in microglia only, or we artificially overexpress SPI1 driven by Rosa locus. And what we find by Western blot is that we indeed achieve about 50% reduction in SPI1 levels by knocking out one allele, which we call a quote unquote knockdown. And by overexpression, we increase PU levels by 30%. And we also confirm this by immunofluorescence. Here you can see a P1 knockdown model where we lose the overexpressing microglia. And here you can see the overexpression model where we have very high expression of P1. And you can see the quantification here. And so far, I will only show you the data from knockdown. However, now we have the overexpression data. We're analyzing it now. We see a very stark differences between knockdown and SPI1 knockdown and wild type in Alzheimer's disease. While in wild type background, these differences aren't immediately clear. So now what we see in these different clusters between SPI1 knockdown and wild type is that if you remember these different molecular signatures we found associated with these three microglia subtypes, what we find is that by downregulating SPI1, we completely abolish this aberrant microglia signature, which we found was associated with higher expression of P1. We have an increase in the signature associated with plaque-associated microglia, and we also have an increase in homeostatic signature of microglia. Currently, we are also doing a single cell level or microglial single nuclear analysis to support our findings at the population level. So well, we wanted to, of course, visually confirm that what we are seeing is happening. So we found that HSP90 could be used as a marker for stress response. And we found that in the F.A.D. SPI1 knockdown cells, we have a rescue of cells that are positive for HSP90. We also found that body-pi positive lipid droplets is a marker of these aged microglia, which could be called neurodegenerative. And we see that in the P1 knockdown model, we have a loss of this lipid droplet positive microglia. In addition, if we look at microglia, how microglia behave around the plaques, we can see that SPI1 downregulation results in an increased number of microglia associated with plaques, and SPI1 overexpression results of microglia kind of being less interested with the plaque, kind of retracting their processes from the plaque surface, where they cover less of the plaque, indicating that SPI1 downregulation really increases this plaque-associated microglia response, whereas SPI1 overexpression might be downregulating it. So they have an opposing function. So, but what could be the mechanism of these change in populational dynamics. Now, if you consider um, exposure to this amyloid plaque, trying to remove it in a maybe trying to remove it by phagocytosis, failing at it, or just increased continuous phagocytosis of these little filaments associated with the plaques. So this is a very strong chronic stressful stimulus for microglia. So this might result in microglia starting to activate stress response to preserve their life and kind of try to return back to homeostasis. So, and then when a cell encounters a stressful stimulus, it has kind of two options. If this that can be dependent on both the cellular identity and the quality or strength of the stimulus. So you can either activate stress response, which appears to have more neurodegenerative outcome, or the cell could undergo apoptosis and be replaced with a new cell, which would be a type of rejuvenation of through microglial turnover. And what we see by SP1 downregulation is that we, at the molecular level, we have an increase in both apoptotic signaling and cell cycle signaling. And we can actually see by using Ki67, we can see that there's an increase in the dividing cells around the plaques when we knock down P1. And we right now are confirming these results by BRDU labeling in vivo and also by cultured microglia, where we see clusters of microglia dividing when we knock down P1 in primary microglia. So, okay, so our results show that by SP1 downregulation, we might be if you consider like an age-dependent change in microglia population, where we lose the protective homeostatic and plaque-associated populations, and then we gain this harmful aberrantly activated microglia, and SP1 knockdown prevents this or delays this conversion. So what happens to neuronal health? What's the, if we block this change in age-dependent dynamics, do we rescue any of the AD-associated pathologies? So as I mentioned, in 5X, in Alzheimer's disease, we have two major pathologies, that is amyloid plaques and tau aggregates in the neurons. And what the 5X FAD model lacks is the tau pathology. So the mice never developed tau pathology. To study the effect of microglia, change in microglia function on tau pathology, we collaborated with David Holtzman's lab, and where they injected tau aggregates into the brains of our mice. And what they have previously shown is that in TREM2 knockout mice on amyloidosis model, APPS1, there's an increase in the propagation of tau. And in our preliminary analysis that is now going under depth analysis, what we see is that there's a significant rescue, even at very low numbers, using P1 knockdown animals. There's a significant blockade of this tau propagation, showing that really really confirming that this aberrantly activated microglia could be involved in the propagation of these tau aggregates. We also wanted to see the effects of protective and harmful microglia populations by looking at the spine density of neurons in the cortex. So to look at this, we have collaborated with Dory Schaefer's lab, where they looked at V-glu2 spines in the cortex. So this is how, here in green, you can see the spines in a normal and healthy mouse with microglia in cyan. What you see in normal 5XFAD mice is that there is complete loss of spines where there are plaques, which is a local damage. There is then there are some aggregates of these either autofluorescent or VGLU2 positive kind of dystrophic areas. And then you can see that if you look at globally, there's a global reduction in spine density. If you look at 5XFAD model, the first two observations remain the same, meaning where plaques are, you don't see any spines. Around the plaques, probably overlapping with the dystrophic neurites, you see large aggregates of spines. But if you look at the global densities, which we quantified here, you can see that there's a complete rescue of the spine density, global spine density by P1 downregulation. So to summarize this part, we identified these two different microglia subpopulations in Alzheimer's disease. And by one is characterized by innate immune functions around this plaque-associated microglia, and the other one is aberrantly activated microglia characterized by stress response and metabolic dysfunction. And we found that this is associated with an increased turnover of microglia, which could be preventing the initiation of the stress response, which is a characteristic of these aberrantly activated microglia. We show that this is also associated with increased neuronal health, supporting the idea that increased strengthening of plaque-associated microglia and homeostatic microglia and preventing the emergence of aberrantly activated microglia could be a mechanism that exacerbates Alzheimer's associated pathologies. Now, to briefly mention what I'm planning in the future, so if we again come back to our original model, we have this age-dependent loss of protective microglia and increase in the emergence of harmful microglia, which is considered, which precedes tau pathology and cognitive decline. So in this progression, genetic risk factors play a significant role. However, around 40% of the cases of dementia can be attributed to lifestyle and environmental factors, many of which are modifiable factors. And here you can see a figure from a recent review where above this middle line, you can see factors that increase your chance of developing dementia. And below the line, you can see factors that protect you from dementia. So what I now would like to study is that what happens to microglia that are exposed to these different types of lifestyle. This could be diet or exercise, or we were just talking with Masashi, it could be polluted air, and many social exposures, which we all experienced through COVID. And how does this change the molecular identity, metabolic state, and epigenetic function in microglia, so that the response of microglia to aging associated triggers such as amyloid aggregates will result in the emergence of these harmful type of microglia. So I would, this is basically what I'll be focusing on in my own lab. And I mentioned some of our collaborators, but both of these studies have been very big collaborations with Dory Schaefer's lab, Meinrad Buslinger's lab, Oleg Butowski's lab, many groups at Sinai, Of course, Anne has been a great mentor, and everybody in Anne's lab, it's a very collaborative lab within itself too. And of course, Musashi, thank you very much. And I'm ready to take some questions.